classes. So this means we are going to look at two different traits at the same time. Two different traits. I'm going to switch to black just because I like that better. So let's take two traits at the same time. And we'll use Mendel's pea plants because it's easy to keep track of. The first trait is going to be color. In color, for pea plants, there are two alleles for color. And we've already used capital Y for the dominant yellow color of the peas themselves, and small y for the recessive color of green. That's trait number one. Then we're going to keep track of our second trait. I'll put it over here. And that's going to be the texture of the skin of the pea. And it's either going to be smooth. So S is a hard letter to use. Even though smooth is dominant, I'm going to use big W to mean smooth and little w to mean wrinkled, because then you can tell the difference between them. Remember, it doesn't matter which letter you pick as long as you have the dominant as a capital and the recessive as the small, and it's a good idea to choose letters where the small and big letters are easily distinguished so you don't get confused. So smooth and wrinkled pea plants. Now, here's what we have to think about in terms of what's going on. Remember, the Punnett squares we draw are just models of what's actually happening in meiosis. So what's happening is that we have a gene here. Here's a chromosome. And here's its homologue, right? The other chromosome that's sort of just like it. And in this particular person, there's a gene. I'll put the gene allele. And the alleles would be at a certain spot here, right? And so that's where the two alleles would come from. So let's take, if we had a, maybe it would be for color, maybe it's a capital Y and a small y. But it could be two capital Ys, it could be two small y's, right? It could be any one of those situations. And we said that what happens in meiosis is these line up and they get pulled apart, right? So then we also have another set of chromosomes. Over here, we have a different chromosome, because remember, in humans, there's 23 pairs, right? 46 chromosomes. So here is this one, and it's homologue. And on this one, there's a different spot for a gene right here, and this might be the gene for wrinkly or smooth skin on the pea. And so maybe maybe it's homozygous. Maybe could be two big W's or two little W's. That's what those little letters are standing for, right? And so when you think about metaphase, both of these pairs are going to line up and they're going to be pulled apart, each to one side, to make the sperm and the eggs, the gametes. And they can flip. There's all different combinations that are possible. So now we have to consider, if this was our cross, this could be one of the sperm cells, if this was the male, and it could go with this egg. But that same sperm cell could also go with this egg. That's the same combination. But what if it was this sperm cell with this egg? Or that sperm with this egg? You could get all the different combinations. And so what the Punnett square does is it allows us to sort of map out all the possible ways that these sperm and eggs could get together, right? Okay. So let's do an example. A homozygous, green, smooth, oh wait, I better add a word there, sorry. Homozygous green, heterozygous, smooth pea is crossed with a homozygous yellow Uh, let's go heterozygous smooth. And what we want are to figure out what are the genotypic and phenotypic ratios of this cross. What are things going to look like, and what are the ratios that go with having different options? Like before, we have to look at our alleles. Okay? So we have to figure out, first of all, what the parents look like. Now, in this question, we're being told enough information to know what the parents are. As you saw before, sometimes what they do is they give you vague information about the parents, more information about the offspring, and you kind of have to work backwards, right? So sometimes you're working backwards to get what you need to get. Okay, so let's go and look at the first P, homozygous green. Well, remember, green is the small recessive allele. And so it would have GG as its genotype for that. But now we're also going to add heterozygous smooth. That means two different alleles. So that means the capital W and the small w. So now we have a genotype that contains two sets of alleles instead of just one. And then the other parent, it says, is homozygous yellow. Homozygous means both the same. And if it's yellow, those must be, whoops, I made a mistake, didn't I? I used the wrong letter. It's not G's. We decided to use Y's. So this would be two small Y's for green. My mistake. And these would be two capital Y's, right? Because both of these, nope, sorry. Where am I? Yeah, two capital Y's. Get this straight. Homozygous yellow. Heterozygous smooth. So it has the same as the other one, heterozygous smooth. So those are the two genotypes that we have. And we're keeping track of two traits instead of one. All right. So now we have to think about the possible gametes. This is where it gets a little bit tricky, because the red chromosomes can combine in any way with the blue ones, depending on which one gets pulled where in meiosis. So the possible genotypes for this person, if we look at the red, there's only one letter they can give. It's a small y. But that small y could end up with the big capital W. That's one possible, let's call this the male, that's one possible sperm genotype. But the small y might also end up with the little w. So the other possible genotype would be this. This would be the other possible sperm you could get. Essentially, you have to take each red one and blend it with each blue one and see how many different combinations you can get. Over here, it's kind of similar, except that the only one we could get on this side is a capital Y combined with the big W. That's one option, right? So you'll notice how our gametes have two letters in them now, but that's because we're talking about two different genes on two different chromosomes. And then we have another gamete that would be, again, it would have to be a capital Y because there's only one option, but cross with the small w. So these are now our sperm and egg cells. They have two letters in them because we're tracking two traits instead of one. But those are not the same alleles of the same gene. That's why I've drawn them in different colors, and that's why we're using different letters for them. Okay, so now we have to draw a Punnett square with the possible gametes. So let's draw our Punnett square. I'll get a nice okay, and then let's go and put them in. I'll try to color code them here. The possible gametes are Y and big W, or Y and little w. Right? There are the possible gametes from this side, this parent. Then I'll take my black and I'll put a little line across. The possible gametes from the other are a big Y, and the other one is a big Y, and then we have a capital W or a little w. Those are the possible gametes from the other parent. And we'll put a cross in here. So we're going to get a Punnett square that has 4 possible or different combinations because we have only got 4 possible gametes that we can produce. And so we put them together. So in the first case, we have a big Y and a little y, and then 2 capital W's. Down below, we have a big Y and a little y, but then we have the capital W and a small w. And then over here, we have a capital Y and a small y, crossed with a large W and a small w. And then the last one is a capital Y and a small y, crossed with 2 small w's. And so now we can look at what this means. These are the genotypes. So the first possible genotype, what we can do is we can make a little chart if we want. We can put, let's make a little chart. I just don't have enough room. I should have moved this over. I'll do it down here. All right? G for genotype and P for phenotype. So my first possible genotype is this one, which is Y heterozygous and then homozygous dominant, like that. What would that produce? Well, if you look at the Y's for the color, the dominant Y would produce a yellow P. So the phenotype is yellow because of the way the Y's turned out. And then the 2 capital W's are telling me it must be a smooth P because that was the dominant situation for the gene. Okay, then we look at the next genotype right here going across. We have small y, sorry, a big W and a small w. Now that genotype occurs twice. It also occurs here. Those are both the same. So we don't have to write it twice. That's just another possible combination. But I guess we should keep track of the possible. This one, the first one, was 1 out of the 4 possible options, right, for the percentages. This one is going to end up being 2 out of 4 because there's 2 out of 4 ways that this will show up. It still means, though, that we'll get a yellow P because it has at least 1 capital Y, dominant, right? And it will also be a smooth P because the capital W makes it smooth, dominant gene. So that means we would have to, if we're going to do the phenotypes, we have to put these 2 together because they're the same thing, aren't they? So these 2 are both the same phenotype. So that means actually 3 out of the 4 options are producing the same phenotype. Even though the genotype is slightly different, they still look the same, right? Because of the dominant genes. And then, of course, we have the last option here, which is going to be, let's put it in here, a capital Y and a small y and 2 small w's. That's going to produce a yellow P because of the capital dominant Y, but it has 2 recessive alleles for wrinkled. And so this will be a yellow wrinkled P. And that will happen 1 out of the 4 times. So the ratios for the genotypes are 1/4 this, 2/4 this, and 1/4 this. But for the phenotypes, it's 3/4 and 1/4 because these 2 right here look exactly the same. Phenotype ratios. So that means that when this plant has seeds and makes seeds in this cross, we can expect about 3/4 of them to be yellow and smooth. And we can expect about 1/4 of them to be yellow and wrinkled. Remember, these are probable, I guess I could say, probabilities. They're not actual numbers. It doesn't mean every time they have seeds, you're going to get 3 and 1 out of every 4. Well, it kind of does, but it has to be over a large number of seeds. Okay? The first 10 seeds you count could all be yellow and wrinkled. But if you count 500 seeds, then eventually you'll see that it will come out to a 3-to-1 ratio for these two phenotypes. Okay, we'll do one more example, just a little bit faster. We'll do sort of the one that's going to give us the most work. And that's going to be something that's heterozygous for both traits in both parents. So we'll use the same P. We'll say that we have parent 1, and that parent is this genotype. I won't color code it anymore because now you get the idea. And we're going to cross that with the other parent, which has the same genotype. Okay, now to get to possible gametes. What we have to do is we have to take the first Y, and we have to combine it with the other possible letters that go with it. So the first one would be Y with a big W, also a capital Y and a small w. But we also then have to take the small y and combine it. So maybe the small y will have small y, big W, and small y, little w. So now there are 4 possible ways to make the combinations of letters, right? As the genes pull apart, as the chromosomes pull apart in meiosis, each one could end up with the other. And it's going to be the same over here. You're going to be able to get the same ones, because this Y go with the big W, but then it could also maybe get caught with the little w in this particular sperm or egg. And then we have the small y with the big W, and the small y might end up with the little w. So what this is it's the law of independent assortment, right? What it's saying is that the y's and the w's get to sort themselves out without any link to each other. Independent assortment. All right, so now we're going to have a pretty big Punnett square because we've got 4 possible gametes for each parent. So when we draw our Punnett square, I have to come down here, draw a nice big Punnett square because we're going to have lots of options. So let's make it like this. So I'll write down the side all of these possible genotypes. And I'll just do it in black. So big Y. Oh, let me get a smaller font or smaller line here. That's pretty much too thick now. So we have actually I'll pick the standard. We have a big Y with, the first one was what? A big Y and a big W, right? And then a big Y and a little w. And then a little y and a big W and a little y and a little w. So I have 4 possible gametes. Same thing on the top. Same thing. So my Punnett square is going to have 16 little squares in it, 4 times 4. Now I have to do all my crossing. So in the first one, I'm going to get capital Y, big W. Actually, you know what I'm going to do? I am going to color code them because it's easier to see later. So I'll put the Y's in red and the W's in blue. There's the first combination. The next combination is going to be this. And then down here, I get this combination. And then down below, I get this combination. I get a big Y and a little y, right? And you might be wondering, is this the same as this? And the answer is yes. It doesn't matter which way you write them. I always put the capital first out of habit. So if there's a capital, I put it first because it just, for me, it just looks right. That one, I can easily confuse that with something else. And I find that writing the small one first is just kind of, doesn't make sense. So it doesn't matter, because you might think, well, maybe the little y should go first. It doesn't matter which one goes first. But out of convention, we tend to put the capital one first and the small one second, just to keep things clear, so it always looks the same to our eye. Because as you can see, we're going to have to track which of these are different, and there's 16 of them, so it can get a little tricky. Okay, let's do the next column. A big Y, a big Y, a big W, and a small w. A big Y, two small w's. A big Y and a small y, a big W and a small w. And then a big Y and a small y, and then two little w's. Okay, and we move over to the next one. A big Y and a small y, two big w's. So remember, what this is doing is it's giving us all the possible ways that the sperm and eggs that have formed could possibly combine, because we don't really know exactly which sperm or egg it is, and we're using all the possible combinations at fertilization. So the Punnett Square represents the fertilization stage. We did the gametes first, that's the meiosis part. Now we're doing what happens when they fertilize to put them back together, right? And so we get all these combinations, and some of them, as you've noticed, are repeating themselves. Okay, two little y's, that's the first time we saw that, and then a big W and a big W. If I make a mistake, you tell me, because it's easy to get going, because I'm talking at the same time as I'm writing, and makes for a more interesting video, but it means I'm more liable to make an omission or a mistake. I think that's good so far. Now, one last column to do. Big Y, big W, little w. Big Y, little y, two small w's. Homozygous recessive. And when you write this, if you're studying, say that to yourself. Say the word homozygous recessive, so you start to associate that and you understand. Like this next one, homozygous recessive for color. Heterozygous for wrinkled or smooth. Heterozygous. The next one is homozygous, because it's two the same Y's. And if you say that to yourself, in a sense, you're actually studying all that vocabulary while you're doing your Punnett Square. There we go. Now what we have to do is put it all together and look at all the possible genotypes. Now some of them are repeats, so we can eliminate a few. Okay? The first one, I'm going to use this yellow color to circle them. The first one is right here. I don't see any other place in all 16 squares where that happens, right? That's the only one. So that would be, if I'm doing my genotypes, so I'll put my genotype here, I'll put my phenotype here, right? Actually, you know what I'll do? I'll put genotype, and I'll put the ratio, R, for the genotype ratio. Actually, I'll go really mathematical here. I'll put G with a little r, genotype ratio. And then I'll put phenotype, which I'm going to have to write lots of words for, so I'll leave a big space there. And then I'll put phenotype ratio, like that. Actually, if I was really being mathematical, I should do that the other way around. Let's be math people. It's a ratio, and we would put the little g to describe it as the genotype ratio. There, we'll use those letters. Okay, so my first genotype, right, is big Y, big W, big W. That was the first one that I circled in yellow right there. And there's only one of those in the whole thing. So 1 out of 16. possible squares. What would it look like? Well, its phenotype would be yellow and smooth. Okay? Now, I'm not going to put the phenotype ratio in here because I might get more yellow and smooths, right? So what I could do is I could put, okay, you know what I'll do? I'll put the 1 out of 16 here. But this is sort of like not, we have to add these up after. So we're going to have to do something with that column before we're finished. Okay, let's look at the next genotype. The next genotype would be, if I go to yellow, let's go this way. 2 homozygous and then heterozygous. Now I'm looking there, but I also see it here. Same thing. So I'm going to put a big X so we can mark them. Those two are the same. I have to look around to see if there's any other spot where they're the same. And there isn't. There isn't any other place where I have 2 capital Ys and heterozygous for the W's. So there's 2 out of 16 now for that. So what I have here now is YY and big W, little w. 2 out of 16. Those phenotypes will also be yellow and smooth. So you see the problem. We have to add these up after. So there's 2 out of 16 for that. Okay? So far, we're just copying the genotype ratio under the phenotype ratio, but later, we're going to have to go back and add up all the ones that are the same. Okay, what's the next combination? Well, let's kind of keep going. Let's look at this one, which is heterozygous for, you know what, I'll use a different color. Heterozygous for the color, homozygous for, what would that be, the wrinkly, smooth. There's one here, so those two are the same, right? I don't see any more. I only see 2 of those options. Okay, so let's write that one down. And that is, what was that one? It was hetero and homo. Heterozygous and homozygous for the dominant, right? Our chart go down a little bit more here. And how many were there? There were 2, I think. There are 2 out of the 16. So we'll go 2 out of 16. But the phenotypes of this are still yellow and smooth. So the phenotype for all these combinations hasn't changed. 2 out of 16. Yellow and smooth. All right. Now we're going to do the next combination. Let me take the green color. Let's do the 2 capital Y's with the 2 small W's right there in the middle. And I think that's the only place where that happens. 2 capital Y's and 2 small W's. Now there should be another place where that happens. Why am I missing it? Let's go back and check. I'm thinking that, no, maybe not. There's only 1 cross where that can happen. Okay, I guess we're all right. 2 capital Y's and 2 small W's. One out of 16. The phenotype for that is going to be yellow, but this time wrinkled. Yellow and wrinkled. 1 out of 16. So far, so good. Now, let's pick I'll go back to my yellow color again. Let's do this one. Heterozygous for both traits. There's one there. There's one here. It kind of goes diagonally. One here and one there. Okay. Now, of course, some of you like to memorize patterns. The pattern that I'm creating here with the diagonal only works if you've written your gametes in the same order. So you can't really memorize the pattern because you might write your gametes in different orders on the sides, and so you basically have to just hunt for them. I don't see any others, so there are 4 of those. Heterozygous both traits. So we write y, little y, big W, little w, 4 out of 16. And that would mean these are going to be, guess what? Yellow and smooth again. So these are the same phenotypes as the first 3. So we'll have to come back and make sure we add up those later to get the proper phenotypic ratios. Okay, and we go down to the next set of things that are the same. I'll go back to my purple color here. Let's do heterozygous Y, homozygous W. One there and one down over here. Those are the only 2. Heterozygous Y. Oh, wait a minute. Yeah, heterozygous Y, homozygous recessive. Heterozygous for this, but now 2 small w's. Homozygous recessive. All right. Essentially, what we're doing here is bookkeeping, right? And there were 2 of them, right? So 2 out of 16. Sorry, green. 2 out of 16. The phenotype would be, again, yellow. So far, everything's been yellow, but now wrinkled again. So we'll have to make sure we add that up with the one before, where we had yellow and wrinkled. 2 out of 16. So far, so good. We're getting to the end. It looks like we have just 3 more combinations. Right here in the middle, there's this one, which occurs only once, as far as I can see. It's homozygous recessive, homozygous dominant. So let's add that one in. Homozygous recessive, homozygous dominant. I've got to double check to make sure that's what I said, because when I'm yep. 1 out of 16. That phenotype will be the first one we've come across that's green, because it has no dominant yellow allele. But it will be smooth, green and smooth. And that will be right here, 1 out of 16. Okay, almost done. Go back to yellow because it's easier to see. And then we can see we have the homozygous recessive and the heterozygous condition. 2 small y's and then big W, little w. 2 small y's, big W, little w. And there were 2 of them, so 2 out of 16. And that phenotype is going to be green, but still smooth, because that one capital W is enough to mask. So 2 out of 16 for those. And then I think we have one more, right? Let me just add some lines here. I think we have one more. And it's the very last option right here. I'll do it in black. It is the homozygous recessive for everything. So we have 2 small y's and 2 small w's. 1 out of 16. And this P will be green and wrinkled, both of the recessive condition. 1 out of 16. So in our chart here, the genotype ratios in green are all correct. But the phenotype ratios, there's overlap. There are some that look the same. So now we have to do the phenotype ratios by sort of summarizing them and adding them together, because these numbers aren't right. So what we have to do is we've got to take this one, yellow and smooth, yellow and smooth, yellow and smooth, and down here, yellow and smooth. There are 4 different genotypes that produce the same phenotype. I don't see yellow and smooth anywhere else. So that means I have to add up 1/16, 2/16, 2/16, and 4/16. And I believe that will be 9/16, right? So 9 out of 16 will be yellow and smooth. So I would write down below here for my phenotypes. So let's put actual phenotype ratios. It would be yellow and smooth, 9 out of 16. Then I go back and I see, okay, what else have I got here? Well, I've got yellow and wrinkled right here. So let me look at yellow and wrinkled here and yellow and wrinkled here. That's 1/16 and 2/16. So that's 3/16, right? And there's no other yellow and wrinkled peas. So 3/16 for yellow and wrinkled. Then I go back up. I've got those 5 already covered. So now we'll look at the green options. And it looks like I have green and smooth here, green and smooth here. 1/16 and 2/16 means 3/16. So I go down here and I write green and smooth. 3/16. And then the only thing left for a phenotype is green and wrinkled, the very last one, and it's down to 1/16. 1/16. Green and wrinkled, 1 out of 16. So I have to add up all these ones that go together. So we have 9, 3, 3, and 1. So that means that if we have 5,000 seeds produced from this cross, that for 9 out of 16, or that ratio, we can expect to be yellow and smooth. We can expect 3 out of 16 out of every 16 to be yellow and wrinkled, 3 out of 16 to be green and smooth, and 1 out of 16 to be green and wrinkled. Remember, these are the probabilities. It doesn't mean that you're going to have 16 seeds. The 16 doesn't indicate the number of seeds. It just indicates sort of the probability. It means for every 16 of those 5,000 seeds, we would expect these numbers. And the more seeds we make, the closer we would get to these actual numbers. And that's the dihybrid cross. Now, because the dihybrid cross is 2, there are lots of different options. Your Punnett squares, some of them will have 16 squares, some of them might only need 12 or 8 or whatever. But there's so many different options, it's a little bit more tedious to keep track of. But that's the process. It works the same way. It's just everything is now working with 2 alleles instead of 1. Okay? So we'll stop it there.